All right, can everyone see that? Sorry, what was that? Okay. All right, guys, we'll get started. All right, I hate to be that guy, but if we can have, it's fine. You don't have to, but if you can put your camera on, that would be great. It allows us to engage a bit more. If not, really don't worry, it's a Sunday. So we'll go over GI and endocrine system. This session, I've made it half teaching, half SBAs. So main reason for that is I started it off being entirely teaching, but you guys from the last session, you liked SBAs. So for the last three lectures, we'll do mainly SBAs, okay, to cover the content. But for the first couple of slides, I'll teach it to you, okay? Throughout the lecture, if you have any questions, if you're unsure about anything, let me know and I'll try and answer it for you. I did this quite a while ago, so I am sorry if I get something wrong, although I doubt I will. And yeah, we'll get started. So we've got 113 slides, but we'll go through them quite quickly. Okay. Yeah, thank you, Kimberly. Let me just let the other chat know. Get started. Okay. Yeah, as I said, any questions, just interrupt me. All right. So first lecture, gut introduction. These are the knobs. By the way, can you guys see my mouse just for reference? Can you see me moving it here? Can you?
Yeah, we can.
Brilliant, thank you. Okay, so I'm sure you guys know this by now, but just to remind you of what parasympathetic and sympathetic system is, the way I like to differentiate between the two, because in first year I found it difficult, is your parasympathetic system is everything. It does everything you don't want to happen when you're running away from a dog or a lion. So to remember that easily, if you're running away from a dog, do you want to wee? No, because it's not going to help you, is it? Do you want to do a number two? No, it's not going to help you. So your parasympathetic does everything you would do while you're in a calm state, while you're relaxed. And that means it makes up the bulk of digestion. So digestion is a very at rest kind of thing. If you're doing exercise or you are running away from a lion or a dog, your digestive system, the blood will be diverted away from your digestive system because your sympathetics will be activated. So it directs the blood to more useful parts where you need it. I hope that makes sense. So the role of the autonomic nervous system. So as I mentioned, your vagus nerve and your parasympathetics are going to be important here. So the stomach, what can it do? It can store up to 1.5 liters of food. Okay. And the way this happens is your fundus, the top of the stomach, relaxes alongside the body of the stomach. Okay. So you can see the fundus in yellow up here, then the body in the middle. Okay. So when you eat a large meal, you have mechanoreceptors, and they sense stretch in your stomach. The large amount of food, you sense the stretch, and this causes your parasympathetic to become activated. Your vagus nerve, then, this is the main parasympathetic nerve. Vagus nerve is involved in everything parasympathetic. You'll get VIP and nitrous oxide released. And what this will do is this will cause relaxation. Okay, so you can accommodate more food, basically. And yeah, it just allows you to store food in the stomach. These two VIP and nitrous oxide, they come up a lot, and they always cause relaxation. So if you remember that, they cause relaxation of different things, you should be fine. And then, so what happens is your vagus nerve, yeah, we mentioned that. the reason the antrum is important, so the vagus will cause contraction of your antrum. And the reason this is important is you want to kind of mix up the food and then also push it towards the pylorus, which is your kind of your gate between your stomach here and your duodenum, which is the first part of your small intestine. So you can see here the pyloric sphincter, and yeah, so. And then the duodenum, what it will do is it can sense when the stomach is passing chyme through the sphincter into it, and it has chemoreceptors, so it can also sense acidity. And when the duodenum will sense too much acidity, too much chyme, it's sort of a negative feedback system. So when it says, "Look, I've had enough," there's too much here, it will send more. It will kind of send signals which mean, we'll talk about this later, this comes up again, don't worry. It will say, I want the sphincter to close, stop giving me some, but I'll explain this later slide. The enteric nervous system, what is it? It's the brain of your gut. You have two plexuses. You have your myenteric plexus, and you have another one, the submucosal plexus. Yeah. The myenteric is kind of the important one for what we're going to talk about here. And it coordinates peristalsis. Okay. Peristalsis is your basically your movement of your food bolus down your GI system. Okay. It's very important for that. And this, yeah, it's how food moves along your system. Okay. If you want to know a fun fact, there's a disease called Hirschsprung. You don't need to know this. There's a disease called Hirschsprung disease, and this is basically where this myenteric plexus doesn't develop properly in very young children, so they can't move food along a certain area of their tract, so they have to have that removed surgically. Yeah, but you don't need to know that. Don't worry. Okay. So yeah, as we mentioned, peristalsis. So the way this peristalsis kind of works is, of course, we want food to move along. So what happens is you need contraction behind the bolus of food, and you need relaxation in front of it. Okay. Yeah, and so your contraction is mediated. So behind, really mediated by acetylcholine, substance P, and the kind of relaxation bit, the inhibitory bit, as I said before, nitric oxide and VIP. So yeah, if you just remember relaxation in front, contraction behind, is relatively quite simple. Yeah, does anyone have any questions so far, or all good? No? Okay. Fine. And yeah, your enteric nervous system and your myenteric plexus, as part of that ENS, is what's allowing this to happen, allowing the food to move along. Okay. So regulation of gastric hydrochloric acid secretion. So you have a few important things here. Your vagus nerve is obviously involved, which cause so the important things are acetylcholine, gastrin, and histamine. Okay, these are your three things which are very important for hydrochloric acid release. So acetylcholine causes this release in a few different ways. So acetylcholine will stimulate gastrin release. Okay, it will stimulate histamine release, and it will also stimulate the parietal cell, the cell which releases HCl itself directly. Okay, so yeah, if you remember, acetylcholine, gastrin, parietal cell itself, and histamine. Okay, gastrin also is involved. This is I've mentioned this in the next slide. Is involved in hydrochloric acid release. Okay, from parietal cell, stimulates it directly. It also acts on the ECL cell and releases histamine as well. Okay, there's kind of 3, 2, 1. Acetylcholine at the top, 3 kind of ways. Gastrin number 2, 2 kind of ways of getting HCl out, and histamine at the bottom. Histamine acting on H2 receptors on the parietal cell brings out even more HCl secretion. So yeah, you have those 3 things which increase HCl secretion, and those 2 things which can both also increase each other, kind of. Does that make sense? Yeah. Okay. So gastrin, yeah, we just mentioned gastrin. Gastrin comes from G cells in the antrum of the stomach, and it's released in response to gastric distension. So what's going to distend the stomach? A full stomach, meaning there's food, there's proteins, so forth, need to be digested. So your gastrins can be released, so you can get more HCl. Okay, and yeah, like I mentioned before, it can directly release, gastrin directly releases HCl, and it also stimulates the enterochromaffin cells to release histamine, more acid secretion. Okay. Quick question: does anyone know? Put it in the chat or say it. What is the thing that decreases HCl secretion? No one? Yeah, there you go. Yeah, well done, guys. Correct. So yeah, somatostatin is released by D cells, and when you have too much acid in the stomach, it's too acidic, your pH is too low, somatostatin will be released, and yeah, that's kind of counteracting too much acid, and it will inhibit the secretion, okay, and suppress gastrin and histamine release. So suppresses all the things that increase the acid release and decrease the pH. So yeah. Okay, here's a table with it all in. I hope that makes sense. Okay, next. Okay, so an exocrine secretion. Okay, so an exocrine secretion is a secretion that doesn't enter the blood. Okay, secretions which enter the blood are endocrine. Exocrine are, say you have an enzyme entering directly into your stomach. That's an example of an exocrine secretion. Okay, and they act locally. So if you enter something into the blood, for example, it's endocrine, that will act around your entire system because your bloodstream is all connected, and it will travel all around your body, essentially. So that's why it's a widespread systemic. Endocrine is usually widespread systemic. Okay, whereas exocrine secretions, they're in a specific place, like the stomach, for example. They act locally. Okay, some examples here. You can read those on the slides later. Okay, so functional motility. Yeah, so the gut muscles move and process food. I think that's quite self-explanatory. They store food. They can mix food with enzymes and bile, breaking it down, and nutrients are absorbed. We're going to go over this later. And then gastric antrum, as I mentioned before, the antrum is partly responsible for grinding up food and degrading it, making it ready so it can enter the duodenum. Peristalsis, coordination of movement. We talked about this. Yeah, you don't want food moving too fast through your system, otherwise you end up with diarrhea, and if it's too slow, constipation. So pretty self-explanatory stuff there. So absorption of nutrients is mainly in your small intestine. Okay, so this is where you get all your good stuff out of your food, in your small intestine. So your glucose, your amino acids, your fats, and the small intestine is really important for that. And then your large intestine is about water. So just to remember, small intestine nutrients and then you'll remember that, oh, the large intestine's the other one, water, or remember it the other way around. So yeah, actually, I will tell you this example because it will come up later. So probably learn it. Glucose. So the way glucose, when it's absorbed in the small intestine, okay, glucose has to get from, so if you picture a small intestinal cell and you have the lumen of the small intestine, you need to go from small intestine lumen, okay, into cell, into blood, okay. So that's 2 movements, 3 places, okay. So you're gonna have different transporters involved in that. I have a nice diagram later I can show you. But so yeah, glucose, for example, is to get from the small intestine into the cells of the small intestine, enterocytes. I think that's, yeah, enterocytes. You have this SGLT1, and then you have a different transporter on the other side, which allows you to get from the small intestinal cell into the blood. Okay, and that's GLUT2 in this example. Does that make sense? Any questions about that? Honestly, any questions, shout out, interrupt me. Okay. Yeah. Okay. Fine. Function of digestion, absorption. Yeah. Yeah. This is self-explanatory, pretty much as well. Why do we digest food? Well, we eat food, then we need to break it down, then we need to take all the good stuff from the food, excrete the waste. That makes sense. And you know that you have enzymes at different parts of your GI tract where they act, and yeah, that makes sense. It's not too difficult. So yeah, you have lots of defense systems along your digestive tract. So your sight, if something looks, if you've ever looked at something and thought, oh, can't eat that, yeah, that's your sight defending you from eating certain things, although it doesn't work on some people, alongside smell and your taste. Something tastes bad, the chances it's probably not good for you. These are all evolved mechanisms in ourselves that we have. So you can vomit, you can chuck up anything bad for you. Your hydrochloric acid in your stomach, this is good for you. It kills bacteria in the stomach. Your microbial barrier, you have gut bacteria. Your gut bacteria are really important. I'm not sure if you guys have heard of the term normal flora yet. They're really important. They compete with any external bacteria coming in, which means that they compete for nutrients, space, resources, so that the harmful bacteria can't survive. You have Peyer's patches, which contain macrophages. They respond to pathogens, antigens, growth factors, and then you have all of, basically you have a lot of different defense mechanisms. Okay, against ingested things that are bad for you. Okay. The gut, you remove waste, unwanted substances. So you can your saliva, you have so when you swallow something harmful, and it can be secreted into the saliva, and then you'll swallow that saliva, it will go down into the digestive tract and be excreted. Okay, bile, break down things you don't want, feces, and then also vomiting is one of the key ones as well. Vomit up anything harmful. Okay. This is what I was explaining before with endocrine and exocrine secretions. Endocrine into the blood, more of a systemic response, although not always a systemic response. Like if gastrin was released, it wouldn't necessarily go and act on your toe, for example. So, but yeah, that's the point. Endocrine, blood; exocrine, into the lumen. Yeah, and we'll go over what these things are. Okay. Any questions so far about that lecture? No? Okay. Is everyone still with me? Give me a thumbs up or something. Great. Okay. Good. Good. Very good. Okay. Next lecture, the liver. Okay. I like the liver. The liver's interesting. Okay. So let's explain glucose and the liver. Okay. So as I was mentioning before, with glucose, of course, you absorb it in the small intestine, like we discussed before. Remember, small intestine, okay, then across a transporter into enterocyte, small intestine cell, enterocyte, and then again into the blood, okay. SGLT1, GLUT2 on the other side to move it into the blood, okay. So what glucose does, okay. Oh wait, hang on. That's GLUT1 into the blood, GLUT2, my mistake, moves glucose into liver cells. Okay, so when glucose moves into the liver cell, yeah, okay. So glucose goes to the liver. What happens when glucose goes to the liver? It's phosphorylated. Why is that important? Because when glucose is phosphorylated to G6P in the liver, it traps it inside the liver, okay? And it stays there, it can't go anywhere once it's G6P, okay? Then, once you have G6P in the liver, has two kind of options, okay? It can enter glycolysis, okay? Glycolysis, I'm sure you remember from biology. What's the point of glycolysis? To produce acetyl coenzyme A, which will enter the TCA cycle, and then you'll produce ATP, okay? And that's your energy from that. That's how we can use G6P to make energy. Or it can enter glycogenesis. So you're going to store up your glycogen. You're going to make glycogen stores for later use, which can later be broken down to produce glucose G6P, which can then be shunted to make energy. And yeah, these kind of the enzymes involved. Yeah, I'll explain that on the next slide. Okay. Any questions about that? That's not too easy, so let me know. Okay. So yeah, as I mentioned, so we go glucose, fructose, galactose, all your sugary products. They are absorbed in the intestine, taken up by the liver, okay, by GLUT2, and converted to G6P, okay. Excess stored as glycogen, and yeah, okay, or it can enter glycolysis, produce energy. Gluconeogenesis. So what is gluconeogenesis? Gluconeogenesis is making glucose from other things. It's producing glucose that we don't have from non kind of fructose, galactose, not from those sorts of things, but from other things. So you can make glucose from lactate, glycerol, amino acids. Okay, and we do this when, say, if I don't eat for 3 days, I would be doing, well, actually, I think something else would go on if I didn't eat for 3 days. But if I didn't eat for maybe a couple of hours, gluconeogenesis would be happening because my body's trying to produce glucose and maintain the glucose level in my blood. Okay. Yeah. Okay. Glycogenolysis, that's breaking down the glycogen stores. So that's when we're dephosphorylating the glucose 6-phosphate after we've broken it down from glycogen and releasing that back into the blood. Or it can enter, so it's important to understand if from when you've broken down glycogen, where the glucose will enter into glycolysis, or will it go into the blood, maintain blood homeostasis, okay? So the important thing here is usually, so the only place where the important enzyme glucose 6-phosphatase is in the liver. Okay, so it can send G6P where it wants, glycolysis blood. However, it will usually send it into the blood. I don't think that's too important. But it usually sends it into the blood. When you're in a fasting state, you haven't eaten, your blood glucose is low, your liver will shunt mainly your glucose into the blood. Yeah, okay. But if this was in the muscle, for example, also not too important, but if this was in the muscle, you don't have that glucose 6-phosphatase enzyme, so you can't produce glucose from G6P like you can in the liver, because remember we said in the liver you can go from G6P to glucose and then shunt that back into the blood. If you're doing this in the muscle, it doesn't work because you can't do G6P into glucose. So the only path that glucose G6P will have is into glycolysis, okay, where it will be converted into pyruvate. Does that make sense? Tricky, I know. Okay, any questions, let me know. Okay. So liver, the fats with the liver, triglycerides are broken down into fatty acids and glycerol. Okay. Then, if we wanted to produce energy from our fats, the fatty acids will undergo beta oxidation, acetyl coenzyme A as a result, and then you know the story: TCA cycle, ATP, energy. And when you have a low supply of carbohydrates, you can get acetate, B-hydroxybutyrate. This will provide you energy. Ketone bodies are a form of energy. Okay, they can work for you in situations where you don't have other options. Okay, the liver as well. So involvement of the liver, and the liver can also makes lipoproteins. So these make lipids soluble, so they can be transported they can dissolve in the blood, be transported around. And then if you have a kind of excess carbohydrates, proteins, all of these things, these are converted to fatty acids and triglycerides. Okay, so and then these are stored so in your adipose tissue, so essentially makes you fat. If you have excess carbohydrates and proteins, yeah, they convert it. Okay, and I think you guys know this. The liver puts triglycerides and cholesterol to form VLDLs, and this also helps with transport in the blood. Okay. Now, something important to note with proteins is proteins are never stored. They can't be stored. Okay, it's not like carbohydrates or fats, triglycerides. Proteins are never stored. Okay, so proteins are broken down in the liver into There's also actually no, I'll go over that in a sec. Proteins, yeah, so they're broken down into amino acids, of course, the building blocks. And then the whole point of transamination and deamination in the liver is you want to remove the nitrogen. Okay, you want to remove the nitrogen, and then once that nitrogen is removed, the amino acid which you've removed the nitrogen from can then be broken down, used for other things: energy, glucose, fatty acids. Okay, and then the removed nitrogen enters the nitrogen pool. I'm not sure if this is in your lecture, so let me know if I'm talking too much. And yeah, essentially it's removed, and it enters the nitrogen pool, ammonia, and then that's removed by the urea cycle. Okay. Another very, very important function of the liver is it makes plasma proteins. Plasma proteins are really important. Albumin's especially important. It provides oncotic pressure, and oncotic pressure is what keeps it draws fluid, blood into your cells. I'm not sure if you've covered that yet, but it essentially stops them leaking out. Okay.
The emetic signals.
Sorry, what was that? What was that? No? Okay. As I said, guys, shout out if you need to. Okay, so liver has many protective functions. It cleans the blood. It breaks down drugs, alcohol. It clears out bilirubin, ammonia, hormones, medicines, and certain medications, aspirin, paracetamol. It also acts as a filter. It filters bacteria, waste, debris, and you're Kupffer cells, which are basically liver macrophages. They destroy bacteria, old red blood cells, toxins. So yeah, that is a function of the liver. It's also a kind of protector. Bile. So what bile does, it emulsifies fat. It breaks down fat into, so you have large droplets of fat with low surface area. It will break them down into smaller droplets which have a larger surface area. Then they're easier to break down for lipases. Okay. And then bile also helps fat digestion, absorption. Okay. And then also your fat soluble vitamins such as vitamin A, D, K. Definitely learn this. This is quite important to know that your fat soluble vitamins are A, D, E, and K. ADEC, relatively easy to remember. Okay. Bile also excretes waste, and it excretes your waste. Okay. Especially bilirubin. Bilirubin dissolves in the bile alongside excess cholesterol. Okay, we'll go over this and why this can become pathological later. Okay, bile also contains bicarbonate, so it can neutralize gastric acid there. Okay. Okay, so bile is produced in the liver. Okay, something quite important to understand is the path of bile, because you get a lot of, later down the line, is you get a lot of pathology here, and there's a lot of different pathologies that you'll understand if you know this kind of diagram. So liver produces bile. It moves down the hepatic ducts here, okay, into the common, these form the common hepatic duct. Okay, what you have on your on the left here is the gallbladder. Okay, what does gallbladder do? It stores your bile and also concentrates it. Okay, adds things, makes it better. And the things that connect after the cystic duct, the common hepatic duct is now called the common bile duct. Okay, so above the cystic duct, common hepatic duct; below the cystic duct, common bile duct. Okay, and then when you need it, for example, if the duodenum signals, oh, I need some bile, can you give me some, please? It will release CCK. Okay, here's my example. So you eat your fatty meal, it will reach your duodenum, and your CCK will become released. And what CCK does is it will say, gallbladder, look, I need bile. The gallbladder will contract. Your bile will come all the way down here, down the common bile duct. Okay, then go down here into your duodenum. Okay, so that will happen when you eat something fatty in particular. Okay, bile pigments and bile salts. Bile pigments are relatively useless. Bilirubin's a breakdown product, also biliverdin. They don't really do anything. Here's my fun fact, which I found quite interesting when I learned, is the reason your bruise is changing color over time is because, of course, clotting, you have cells there. The cells break down. What do they release? They release initially, okay? They release initially biliverdin. Once that's green, that reduces the green color, and when that changes into bilirubin, your bruise becomes yellow. So yeah, if you, next time you have a bruise, you look at it and you say, oh look, here's biliverdin. Bile salts, these are the useful things. These actually do the bile fat emulsification. Okay, they make up a high percent of the bile, but still very little. Okay, and they're made from cholesterol. and you need bile salts. They're important. If you don't have bile salts, you'll get these awfully smelling poos, and really I've seen one on placement. They smell terrible. So jaundice. So jaundice is yellow skin. It's basically too much bilirubin in the blood. Okay. So if you have too much, for example, we know when a red blood cell breaks down, what's released? Bilirubin. Okay. So if you have a hemolysis, a neonatal jaundice, maybe it's likely due to be hemolysis, Rhesus, that type of thing. But you don't need to know that. Basically, jaundice is yellowing of the skin due to excess bilirubin. That's what you need to know. Okay. Now, the liver also has a function in conjugating bilirubin. This means what it's supposed to do is we want bilirubin to be water soluble, okay? So we can excrete it properly. And what it does, sorry? Accent. Okay. So yeah, what it does is we want bilirubin to be water soluble. What we do, we add glucuronic acid to the bilirubin. That makes it water soluble, so it can be excreted. In the bilirubin is what makes both your urine yellow and your stool brown. That's what gives that color, is because of bilirubin. Okay. So if you say in your exam you're given an example, a patient has come in, they have very pale stool, it's not very brown, and their urine is pale, there's no color to their urine. Okay, do we think that's going to, well, I'll put that, do we think that's going to be unconjugated unconjugated jaundice or conjugated? I mean, I've just showed you guys the answer, so if you get this wrong, put it in the chat or say it. So yeah, if there's no color in the stool and the urine is pale, no color, is that conjugated jaundice or unconjugated? What was that?
Is it conjugated?
Conjugated. So it's, yes, correct. Yeah, it is conjugated. Do you know why? No pressure. No. Yeah. Okay. So it is conjugated because, so I'll go over it and then it should make sense and I'll explain it. Okay. So unconjugated jaundice, okay, you have too much bilirubin before it reaches the liver. This is likely due to a breakdown of your red blood cells, okay, hemolysis, a kind of pathological hemolysis in a young child, or even older people, kind of a blood disorder, which means that your red blood cells are being broken down when they shouldn't be. Okay, you don't need to remember that, but remember, if it's unconjugated hyperbilirubinemia, it's usually because there's a red blood cell problem. Okay, a conjugated jaundice, a conjugated high bilirubin, is usually because there's a blockage. You remember this tree that I showed you? Remember this tree? Okay, there's usually a blockage anywhere here because this it's being conjugated, but now it's building up. Okay, and now it's building up and it's not getting into the urine, it's not getting into the stool. when it should be. Okay, so there's a blockage here, maybe a, I don't know, a stone or something is blocking that. So yeah, bile duct obstruction or hepatitis, so your liver isn't functioning properly. Just anything after the liver means you'll get that pale urine, you'll get the stool issues. Okay, and that's because bilirubin cannot leave the liver or the bile ducts. Okay, so yeah, I hope that makes sense. Hang on. Hang on. Yeah, sorry, you won't get pale urine, you'll get dark urine. I've confused that there. It's dark urine and pale stool. So conjugated high conjugated bilirubin, conjugated jaundice, you'll get dark urine. Because you essentially, yeah, it makes sense because bilirubin isn't entering your urine to give it the color it should have. Okay. So dark urine and pale stool. Sorry, my mistake. It's not pale urine. Okay. Does anyone know what that is? Any wrong answer, it's fine. I know it's Sunday. Any guesses? No? Yeah, gallstone. Well done. Yeah, excellent. Well done, guys. Good. That's a gallstone, okay? You have two types of gallstone. You can have a cholesterol stone or a pigment stone. The way you get a cholesterol stone is if you're eating too much fatty things, okay? Usually in obesity as well, okay? So your cholesterol is, what cholesterol does, I'm not sure if I mentioned it earlier, cholesterol enters into the bile, okay? Cholesterol is dissolved in the bile. If you have too much cholesterol, okay, it will saturate your bile, and then your bile is like, look, I can't take any more cholesterol now, go away. So it precipitates out of the bile, and that's when you get stones, cholesterol stones. Pigment stones. Pigment stones are when you have mainly bilirubin in your stone. That's what a pigment stone is. You have too much bilirubin, okay? And bilirubin will combine with calcium, and that will make your pigment stones, okay? And then, as I mentioned, you have these kind of issues, like you have a hemolysis, breakdown of red blood cells, liver cirrhosis, liver damage, so you can't conjugate the bilirubin. These are the kind of things that lead to your pigment stones. I think if you know the difference between these two, you should be okay. And just know that cholesterol stones are a lot more common. Okay. So the liver, I'm not sure you learn about clotting factors much yet, but it makes most of the clotting factors, almost all of them. 1, 2, 5, 7, 9, 10, 11, 12. Okay. It also makes vitamin K. No, it's the site of vitamin K. And vitamin K is very important for your clotting factors to work, for your coagulation system to be working. It's why, does anyone know, I was going to ask a great question there, but I've just realized I've put it on the slide. I was going to say, what drug inhibits vitamin K, but don't worry. Okay, it also makes fibrinogen, and it also makes liver, makes protein CS and antithrombin. Okay. So yeah, liver is important in coagulation, is the main thing. It makes almost all the factors important for vitamin K and protein CS and antithrombin. Okay. So we're on our last lecture, which will be done via teaching, and then we'll do SBAs, which I'm sure everyone would enjoy more, to be honest. Okay. So we've got a few more slides, and then it'll be pure SBAs. So we mentioned peristalsis, right? So actually, okay, does anyone want to explain how peristalsis works? You can type it or say it, up to you. But we talked about it briefly before. But how does it work? What happens behind the food bolus and what happens in front of it, and which neurotransmitters are involved where? No one? Okay, that's okay. No problem. We'll go over it again. Okay. Yeah, good. Yeah, contraction behind. Yeah, and relax in front. Yeah. Yeah. So just to clarify, peristalsis can happen anywhere from your esophagus. So anywhere essentially your food is moving down all the way up to kind of the entrance of the stomach, or it also happens in the stomach. Okay. So the whole point is if I swallow food here, the way I get my food from my esophagus down all the way to the point into the small intestine, then the way I do that is peristalsis. It's the whole way across. For each food bolus I have, my parasympathetics will be activated. My vagus nerve will be allowing acetylcholine to cause contraction. So say my food's here, contraction will happen here behind the food. And then also in front of the food, so down here, food's here in the middle, by the way, is relaxation, nitrous oxide and VIP. Okay. And it will move along that way. That will keep happening. Circular muscle contraction, relaxation, all the way along. Okay. And that's how your food moves. Okay. How can we access the slides? There's a feedback form I need you guys to do at the end. Oh, hang on. It just reminded me of something important. Basically, when I show you the feedback form, don't do it yet. I need to put a section where you write your email, and then you'll write your email in there, and I'll send the slides to everyone. But remind me at the end to edit the form so you can see so you can type your emails. Otherwise, I won't know who to send it to. So thank you. Remind me to do that. Okay. So yeah, this is just what I was explaining. Bolus moves along. That's how it works. Contraction front, relaxation behind. Any questions there? Okay. Reciprocal control. This lob, yeah, I don't know why they gave it to you. It's literally what we talked about. It is peristalsis. This is how it works. Okay, that's what it is. Okay, let's have a look. Okay, so what gastric emptying is, is as I showed you before, you have stomach, antrum, pyloric sphincter, which is your gate between your stomach and your duodenum, and then to opening the pyloric sphincter will allow food from the chyme from the stomach to move from the stomach to the duodenum. Okay, so that's what gastric emptying is. It's emptying chyme into the duodenum. Okay, and the duodenum is referred to as the first part of the small intestine. Okay, and it's where your first bit of absorption happens, so forth. Okay, so it receives chyme from the stomach, and the duodenum mixes that chyme from the stomach with bile, digestive enzymes, bicarbonate, and prepares it for absorption later down the line. Okay, and it works by a negative feedback system. I'm not sure if you have understanding of negative feedback systems, but I'll explain. So antrum C, where the final mash happens, a bit of a mash in the antrum, then the pylorus that has got the sphincter in there that will open, okay, and then into the duodenum. Okay. So when the antrum contracts, aka it's contracting, it's breaking things down, it's doing that. What that also does is it will inhibit the pyloric sphincter. Why do we want it to be inhibited? Well, because we want it to relax. If it's contracted, it's closed. But if it's inhibited, it's open, it's relaxed, we can move chyme through it. Okay. because that's what we want to do here. We want chyme to move from the stomach into the duodenum. Okay. So that's what happens. So, and then, as I mentioned before, what are the things that always cause relaxation? Nitrous oxide and VIP. So that will relax your sphincter. Chyme can move through. Okay. And then once you have that chyme hit the duodenum, what will happen is this is where this is why we call it a negative feedback system. Your acetylcholine and your substance P will then be released. And the reason for this is because the duodenum is now saying, okay, I've received the chyme, close the sphincter, I don't want any more. And it will contract. Okay? And until the duodenum is finished with the chyme it has, it won't happen again. That's what negative feedback is. It's saying, I have enough, I don't want any more. I'm telling you, don't give me any more. Okay? It's a system where, yeah, I hope that makes sense. Okay. And then secretin and CCK are released. CCK, of course, will allow your bile to enter the duodenum. Why that acts on the gallbladder, brings bile down via contraction, cystic duct, common bile duct, down, duodenum. Okay. And your secretin will also be released. Okay. Yeah. Okay. So I hope that makes sense there. Okay. There were 2 slides away from SBAs, don't worry. Okay. So this is a very easy slide to understand. Carbohydrates are broken down the fastest, then proteins, then fats. Fats are slow because they need emulsification. This takes a longer time. So yeah, this is why fats take a longer time to move. You may feel more bloated after a particularly fatty meal because it takes a longer time to move along the system because there are additional steps needed to break down fats. Okay. And then your carbohydrates are the fastest, then proteins. Okay. Liquids they just shoot right through. They don't need breakdown. They don't need anything. Liquid will move right through and won't take up any time. Okay. But solids need to be ground up by the antrum. They need to be broken down a bit, prepared for the duodenum. Okay. Now, just to what does hyperosmolar chyme mean? Chyme is basically what your stomach is producing. It's like, I don't know how to put it in my own words. Chyme, it's like it is what is produced in your stomach as a result of everything that's in there. And it's kind of it's hyperosmolar means it's very when you have high osmolarity, there's a lot of sodium. Let me have a look for you. Actually, I want to give you a proper definition. Can't really describe it. So it's highly concentrated, so it makes sense. High concentration, mixture of partially digested food that rapidly enters the small intestine. Okay, so highly highly concentrated mixture of partially digested food. Okay, I hope that answers your question. No worries. Okay. Yeah, and then I hope this slide makes sense. Okay. Segmentation. So what's segmentation and the difference between peristalsis and segmentation? So we discussed peristalsis, moving the bolus through. Segmentation is where it's instead of moving along, it's back and forth, and it's also a lot faster. There's more contractions over a certain period of time. Okay. And the point is it's not really to move food along. It's meant to be for mixing. So it's like it's mixing the digestive enzymes and so forth. And it actually, sorry, no, that 1 second. I'm just wondering if it happens in the stomach as well as the small intestine. Yeah, okay, it does kind of happen in the stomach, but that's not. Remember, segmentation is what happens in the small intestine, sorry, not the stomach. Okay, that's my mistake. So peristalsis, moving things along into the GI tracts, into the stomach, and segmentation happens in your small intestine, much more rapid contractions. Your MMC is like a wave. Okay, I understand this. Your MMC is like a wave after you've kind of moved your food along. It just, the best way to describe it is like, for example, say you have a bunch of rocks on a beach, and then the wave comes, right? And let's call these rocks bacteria, waste, residual food. Let's say that's what they are. And then a wave comes and washes them away. Okay. And now they're gone. And now it's clean for bacteria, rocks, waste. That's what MMCs are. They're just cleaning things out afterwards. Okay. Right. Now we'll do SPAs.